Hello and welcome to this A-level biology revision video on carbohydrates. Carbohydrates is one of those topics in A-level biology that comes up in a lot of different questions and covers a lot of different areas. So in this video I'm going to aim to cover some of the main areas that you will be asked about. A carbohydrate is any of the large group of organic compounds occurring in food and living tissues and includes sugars, starch and cellulose. They contain hydrogen and oxygen in the same ratio as water, which is 2:1, and typically can be broken down to release energy in the animal body. Monosaccharides are the simplest forms of sugar and are usually colourless, water-soluble and crystalline. The general formula can be H2N. The most important monosaccharide is glucose, which is a hexose sugar, so it contains 6 carbons and has a formula of C6H12O6. It forms a 6-sided ring as shown below. It has many isomers with the same formula but a different structure, including fructose and galactose. Disaccharides are double sugars and are essential to monosaccharides joined together via a glycosidic bond, which involves the formation of water. This is called a condensation reaction. The reverse, which is hydrolysis, involves the addition of a water molecule to break the bond. Maltose is a glucose 1,4-glucose. It is formed on digestion of starch by amylase because this enzyme breaks the starch down into 2 glucose units. Brewing beer starts with malt, which is a maltose solution made by the germination of barley. The structure of maltose is shown below. So you can see in between the 2 glucose molecules there is a bond formed where water has been removed. Sucrose is a glucose 1,2-fructose. It is common in plants because it is less reactive than glucose and it is their main transport sugar. It is the common table sugar that you put in your tea. So you can see that sucrose is made up of a molecule of glucose that is bonded to a molecule of fructose. Lactose, or sugar that we find in milk, is a galactose 1,4-glucose molecule. It is found only in mammalian blood and is the main source of energy in infant mammals. Right, polysaccharides are basically long chains of monosaccharides joined together by glycosidic bonds. So starch, so the monomer is alpha-glucose, plant storage and insoluble, and so it does not change in water or does not change the water potential of cells when it is produced in plants. This is important as starch does not change the osmotic balance. So it is a mixture of amylose and amylopectin. It is insoluble and compact and can be stored easily because it compacts together. The monomer for cellulose is beta-glucose. Straight chains linked by hydrogen bonds hold it together. Special cellulose enzymes are required to break down cellulose. In humans, we do not have this enzyme, and in many animals, in their guts, they have bacteria within them that produce the enzyme that breaks down cellulose. Cellulose forms very strong bonds and is used for cell walls in plants. Glycogen is a way that the body stores glucose in the liver. Its monomer is alpha-glucose. It's similar structurally to alpha-pectin So it's a polyglucose 1,4. It is made by animals as a storage polysaccharide found mainly in muscle and liver. Because of its numerous branches, it can be broken down into glucose very quickly. So it can be hydrolyzed very quickly. It also does not affect water potential. So finally, the last thing that we need to consider when thinking about carbohydrates is or are their ability as energy storage. So glucose units contain lots of bonds that can be broken down to release energy during respiration and create ATP. The breakdown occurs in a series of steps which are driven by shape-specific enzymes. In plants and animals, only glucose can be broken down in respiration, as only the enzymes which fit its shape are present. Alpha glucose can form long chain with thousands of subunits called amylose molecules. Glucose units are bonded together by condensation reactions, forming 1,4 glycosidic bonds. Amylose molecules tend to form coiled spring-like structures due to the way in which the glucose units bond, making them quite compact. Large molecules such as amylose differ from glucose because they are not soluble in water. However, they can be broken down easily to release the alpha glucose, which can then be used in respiration. Right, make sure you have a go at doing some questions about carbohydrates, and if you're watching this before doing your exams, good luck.